Hi, everyone. Thank you for joining us today. My name is Nilizanis, and I'm the product manager for Stunner at Unchained Labs. I will be your moderator. We will have a Q&A session at the end of the presentation. To ask questions, all you have to do is click on the Q&A in the Zoom navigation bar at the top or bottom of your screen and type in your question. When submitting a question, please avoid clicking the anonymous button so we can reach out through email if you aren't able to get to your question, but we will get to as many of them as we can. And now I'd like to introduce Ross Walton, our Senior Application Scientist at Unchained Labs. Hey, Ross. Today, Ross will talk us through how Stunner has your back by detecting aggregation and quantifying protein simultaneously, and how you can use it to quickly determine the drug-to-antibody ratio of your bioconjugate. And I'll hand it over to Ross. Ross, I'm excited. It's been a couple of months since we've done this together, so I'm happy to be here again with you.
That's awesome, Ellis. I'm excited too. It's good to see your face. I've had my coffee, so we're ready to go.
There you go. I'll hand it over to you then.
Great, thanks. So thanks everybody for being here. So I'm going to talk to you about how you can sound the alarm on protein aggregation using Stunner. So I don't know about you, but I've accidentally set off the smoke detector in my kitchen a bunch of times. But that's actually a kind of a good thing, because smoke detectors tell you when there's an issue that's going on around you that might be easy to otherwise kind of easy to ignore. And since you know about it, it means that you can take care of that issue before it becomes a serious problem. Now, Stunner does something really similar, that is, alert you to a problem very early on, but for protein aggregates instead of for smoke. The Stunner is the first and only instrument that has both full spectrum UV-vis absorbance detection and dynamic light scattering, or DLS, all in a single box. The combination of these two detection methods provides a really in-depth analysis of biologic samples. So you can determine concentration of a protein or an antibody, the size and the polydispersity of the sample, and polydispersity and size tells you whether or not that protein sample has aggregates. Stunner also includes a homebrew function, which lets you build your own custom applications to do things like, for example, determine the drug-antibody ratio of an antibody-drug conjugate, which I'll be showing you how to do, which I'll be showing you some results from an experiment like that in a little bit. Stunner's SBS format plates contain microfluidic circuits that are laid out in the same way as a 96-well microplate. These circuits protect samples from cross-contamination or from evaporation. All you have to do is drop your sample into the input well, and then it's pulled by capillary action into the microfluidic reservoir. Once this plate is loaded into Stunner, the sample is pulled from that microfluidic reservoir into those two fixed path length microcuvettes, which you can see circled. It's these two microcuvettes that makes the high precision and wide dynamic range of Stunner possible. Because of these microfluidic circuits with their fixed length microcuvettes, the UV-vis concentrations and DLS sizing from Stunner are obtained from very small sample volumes, only 2 microliters. There's also no need for any sort of additional sample prep or additional dilution. And Stunner runs are actually finished really fast. If you're just measuring concentration, a full plate of 96 samples takes about 10 minutes. And if you're doing a DLS along with those UV-vis measurements, both are done in about an hour. Stunner has a dynamic range from 0.03 to 275 OD, which is about equal to 0.02 up to 200 mg per mil for a typical monoclonal antibody. It reads the full UV-vis absorbance spectrum for each sample. So, and I'll show you how you can use that in a little bit to determine both the DAR of an ADC or determine degree of labeling of a labeled antibody. When determining the protein concentration from a UV-vis spectrum, you use the absorbance at 280 nanometers, or A280, and a protein's E1%, which is a mass extinction coefficient. Now you can see from this dilution series of IgG that the results are extremely linear and highly reproducible across the entire range of concentrations with an R squared of 0.9999. This gravimetric dilution series of IgG in PBS shows off Stunner's high accuracy and precision. The nearly one slope of the trend line, and again, high R² value between the known concentration and the measured concentration shows their close agreement, as does the actual values shown here in this table. Stunner's measured concentrations are within 2% of the known value, and the percent CVs, which you might be more familiar with as relative standard deviation or RSD, are within 1%. And that's across the entire dilution series from 200 mg per mil all the way down to 2 mg per mil. And that's all thanks to those two fixed path length microcuvettes. And remember, you only need 2 microliters of sample to get results that look like this. So here's how you can use DLS plus concentration on protein samples and how much more info that gives you about a sample. So this sample over here in blue is a 5.9 milligram per milliliter sample of a monodispersed protein. The DLS results show a single well-defined peak, which lets you know the sample is homogeneous and doesn't have any significant aggregation. If you look at the green sample, however, you can tell from the UV-Vis that it's 1.1 milligrams per milliliter concentration, and it has 2 distinct peaks in the DLS result, indicating that this is a heterogeneous sample and that it does contain aggregates. So now I'll take you through a few examples of how some of our customers have used Stunner in the past. First, we'll show you how it can be used to screen formulations, then how it can be used to look at ADCs, antibody drug conjugates, and then finally, I'll show you a case study that we did on how you can use Stunner to evaluate different conditions during process development and optimization for an antibody conjugation reaction. So let's start off with looking at formulation. So you can use DLS and UV-Vis combined to do a really quick screen of a formulation and determine if that particular formulation has caused an antibody or a protein to aggregate. Essentially, what we're really asking here is which of these 2 formulations is better, green or blue? So here we've got an example of a monoclonal antibody at about the same concentration, 50 milligrams per milliliter, in 2 different buffers. It's commercial buffer and an acidic citrate phosphate buffer with some sodium chloride. Now you can see right away from the DLS intensity distributions that one protein is monodispersed, or that the antibody is monodispersed in the commercial buffer, but it aggregates in the other buffer. But we can actually do a little bit of quantitation on that as well. So let's take you through a few of the parameters that you get from DLS results. So first we have the Z average diameter, which is the average hydrodynamic diameter of all of the particles in a solution. We also get PDI, or the polydispersity index, which is related to the ranges of sizes of particles in solution. If there's only one size of particle in a solution, PDI is quite low. And if there's lots of different sizes of particles in a solution, it's higher. Monodispersed samples have PDIs less than or equal to 0.1. So this antibody here, so the antibody in the commercial buffer has a PDI of 0.08, and you can see from the picture, monodispersed. And you can also see from the Z average diameter that the average size of this antibody in this buffer is about 6 nanometers. Now, polydispersed samples tend to have PDIs greater than 0.2, like that sample, and that's like the antibody in this citrate phosphate buffer. You can see quite a bit higher than 0.2. And you can also see that the Z average diameter of the antibody in the acidic buffer is higher than that in the commercial buffer, which is another indicator that, yes, the sample is definitely aggregated. The 2 peaks in the DLS intensity distribution of the antibody in the acidic buffer shows that there are 2 populations of particles in the solution, one with an average size of about 20 nanometers and the other with an average size of about 275 nanometers. Now, it's actually possible that both of these peaks represent aggregate populations, since they're both larger than the single peak in the commercial buffer at 6 nanometers. This is a ton of information. You can imagine how it could be used as part of, say, a formulation screen kind of like this one, but with way more samples. But there are other things you can use it for as well. So Stunner's UV-Vis quantification and DLS aggregation detection is actually great for looking at even more complicated biologics than just a monoclonal antibody, like, for example, an ADC. So When you conjugate a monoclonal antibody with a drug to make an ADC, you have to start thinking about that drug-antibody ratio, or DAR. So with the cysteine linkage, like you'll sometimes see with ADCs, you get a kind of a mixed group of antibodies where the DAR, where the number of drug molecules per antibody molecule, can be anywhere between 0 and 8. And then when you measure the average DAR, it's going to be somewhere in that range. Conjugating antibodies also can change their surface properties, especially if the drug or if the conjugate is hydrophobic. So ADCs can have very different aggregation behavior than their monoclonal antibody precursor. So with ADCs, there's a lot more going on. And so you really do want to have some quick, easy answers for concentration, for DAR, and for aggregation behavior. And Stunner really helps figure all of those things out with this all at once. So for our purposes, for our experiments, we used an ADC mimic that is a combination of a monoclonal antibody reference material from Sigma called SigmaMab that's been conjugated to a dansyl fluorophore via an LC-SMCC linker. Nice thing about using this ADC mimic is that we can compare the ADC mimic to its conjugate-free control, SigmaMab, for all of our experiments. It's also nice because the DAR for this ADC mimic has already been determined using mass spec, and that value gets listed on the COA. Like all antibodies, SigmaMab has an absorbance peak at 280 nanometers, while the dansyl fluorophore has an absorbance peak at 335 nanometers. With Beer's law, you can actually determine the concentration of the antibody based on the A280 value, and then the drug concentration based on the A335 value. And that's actually all you need to calculate an average DAR using the homebrew application in Stunner. And here's the results from one such test. So once we dissolved the lyophilized powder of both the ADC mimic and the SigmaMab, they had close to the same antibody concentration, around 2 milligrams per milliliter. The DAR of the ADC mimic, as determined by Stunner, was 4.17, as you can see both in the bar graph and in this table. And then the average DAR based on the sample certificate of analysis, as determined by SEC mass spec, was 4.2. So extremely close results. With Stunner, you can measure antibody concentration and determine DAR from a single read and read 96 samples in about an hour. Plus, you get DLS results, which I'll show you. It can be extremely important. So here, we diluted SigmaMab and the ADC mimic into either PBS or into that same acidic citrate phosphate buffer as we showed you before. Based on these DLS results, you can see that only the conjugated antibody aggregated in that citrate phosphate buffer. The unconjugated control was perfectly happy in the buffer, and both of the antibodies were monodispersed in PBS. So that really just goes to show you how differently the colloidal stability of an ADC can be from its antibody precursor. So now you've seen how Stunner can be used to look at finished samples. Let's take a look at how you can actually use it as part of process development, in this case of a labeling reaction, the labeling conjugation reaction. So as a model of conjugation, of antibody conjugation, we decided to use FITC, which is a fluorescent label. That's common, and fluorescently labeled antibodies, of course, are common in fluorescent microscopy, used quite often. So for this reaction, first we have an incubation step of the antibody with a significant molar excess of the conjugate. After the incubation, we have, of course, conjugated antibody, but we also have a lot of excess unconjugated, unreacted conjugate left, conjugating reagent left, which actually needs to be removed by a purification step before we can actually look at the final conjugated product. But actually, there's something else to think about here that we're not showing in this picture yet. So aggregates can actually be present pretty much anywhere in a process like this. They can be in that initial starting material, or they can form either during the incubation or during the purification steps. And aggregates can actually be made out of either conjugated or unconjugated antibodies. And they can actually really significantly impact the function and quality of a labeled antibody product. Optimizing a conjugation reaction really should involve determining what conditions minimize aggregation while still reaching that desired degree of labeling. So for our labeling experiment, we tested two variables and used 3 key parameters as outputs. We incubated the antibody either with a 20-fold or with a 40-fold molar excess of the conjugation reaction. We also tested the 20-fold molar excess reaction with 2 different incubation times, either 1 hour or 2 hours. For the outputs, we looked at antibody concentration before and after the reaction to do a percent recovery. And then we also looked at the degree of labeling, or DOL. Now, DOL is the amount of a fluorescent label or conjugate that we have per antibody. And it's actually very similar or analogous to the drug-antibody ratio of an antibody-drug conjugate. But since we're using a protein label, it's not necessarily a drug, it's called something slightly different. The last thing that we looked for is, of course, aggregation, both before and after the reactions. So here we actually have the full UV-Vis spectrum of the monoclonal antibody alone in green. You can see its nice peak at 280 nanometers and then flat across the rest of the absorbance spectrum. And then we've also on here have the 1 hour incubation step in blue and the 2 hour incubation step in yellow. So like the ADC mimic, you can see the 2 absorbance peaks, one at 280 nanometers for the protein, but another one at 495 nanometers over here, which is the absorbance maximum of the conjugate FITC. Now we can see these absorbance, we can use these absorbance maxima just the same way we did with the ADC mimic in order to determine antibody and conjugate concentrations and then look at the ratio between them. Now you can probably see just by looking at this that 2 hour incubation step is going to have a higher degree of labeling, the higher DOL, but let's actually quantify things before we start jumping to any conclusions. So both the reactions with that 20-fold molar excess of reagent had the same percent recovery at about 90%, shown by those green bars. But the reaction with the 40-fold molar excess had a significantly lower percent recovery at 77%. The degree of labeling of this 1 hour incubation, the first reaction, was 0.71, which actually indicates that a significant amount of the antibody remained unconjugated and therefore isn't really useful for doing fluorescent microscopy and actually might be detrimental to that process. Increasing the reaction time to 2 hours or increasing the mole ratio of the reagent increased the degree of labeling. But only the 2 hour incubation also had a good percent recovery, which is of course the other parameter that we wanted to optimize. So that 20-to-1 mole ratio for 2 hour incubation had both the highest, a good yield and a good degree of labeling. So this is kind of, at least of these 3, is the most optimal set of reaction conditions. But there's more we can actually look at. It's just looking at UV-Vis alone doesn't really give you the complete picture of what's been going on with these samples. So Stunner DLS gives us information on the colloidal stability of the samples after labeling. So by comparing the intensity distributions of the samples, we can see all 3 reaction conditions, here, here, and here, caused some small amount of aggregation, peak up here in that 100 to 1000 nanometer range. However, this 40-to-1 reaction condition caused, obviously, the most aggregation. Now, DLS is very sensitive, and the intensity of a DLS peak is proportional to the diameter of the particle to the 6th power, which means the DLS detects tiny amounts of large particles. So if there's any large particle in a solution, DLS is going to tell you that it's there and it's going to show up in that intensity distribution. So since we already know, based on the UV-Vis results, that the 20-to-1 for 2 hour reaction time had the best combination of yield and degree of labeling, we should take a closer look at this sample and see how much of a problem that aggregation peak actually is. So along with those intensity distributions, DLS also provides a mass distribution down here in blue, which describes the relative proportions of different sizes of particles in the solution based on their mass. Basically, how much stuff is of one size versus another size. And remember, large particles scatter significantly more light than small ones. So they're more strongly represented in the intensity distribution than in the mass distribution. So even though the intensity distribution of this 20-to-1 2-hour reaction time has 2 peaks, the mass distribution only has a single peak. This indicates that the aggregates make up only a tiny fraction of the total amount of protein in the sample, but you can still detect it using Stunner. So all this gives you an idea of the colloidal stability of your sample as you tweak the experimental design of your conjugation reaction. And sometimes adding too much of your reagent causes detrimental reaction, or causes detrimental aggregation. And sometimes it causes maybe only slight aggregation or no aggregation. And if there's only a little bit of aggregation, perhaps it could be filtered out or dealt with in some other way. So adding Stunner to your toolbox, like I've shown you, gives you some more tools and more ways of quantifying proteins, as well as conjugates, either a fluorescent label or a drug. It also gives you ways of detecting aggregation as part of a formulation study or during process development. And that's just 2 examples of places you can use Stunner, and where, frankly, Stunner is useful anywhere you need to look for aggregation. And really the big point of Stunner that I think is the most important is that it helps you identify problems before they get too big, and does it from just 2 microliters of sample. So Stunner helps you put out aggregation fires before they get any worse, so all you get is the smoke and none of the action, none of the flame. I hope you guys enjoyed my talk, and I'll be happy to take any questions.
Thank you, Ross, for taking us through how Stunner can help in actually so many ways with its amazing combination of UV-Vis and DLS. We already have some great questions that have been submitted, a couple of questions in the chat as well. So I would like to remind you that please, if you want to ask a question, please use the Q&A section in the Zoom navigation bar. Ross, are you ready for this?
Sure. Yeah, let's go.
Okay. What is the concentration range for Stunner?
So the measure range for Stunner in that UV-Vis spectrum is from 0.03 to 275 OD. Now that the concentration range is actually going to depend a lot on your molecule. So for a monoclonal antibody, that range is from about 0.02 up to 275 OD. But other proteins absorb different amounts of light at similar concentrations. So it'll vary depending on exactly what the extinction coefficient of the protein is.
Okay. Can Stunner quantify non-purified samples?
Yes, it can. So we actually have a specific suite of applications called our unmix applications that can be used to quantify proteins or nucleic acids, so either DNA or RNA, even from unpurified samples when there's the potential for some sort of other impurities floating around. So unmix, really great for looking at unpurified samples.
That's true. Can you use this instrument for other UV-Vis applications?
Absolutely. I mean, like I talked about, UV-Vis has been unmixed applications. Also, UV-Vis can be used to quantify nucleic acids, other proteins. But one application that's pretty unique, that's pretty specific for Stunner, is our KDB22 application, which I think is particularly relevant for people who are looking at proteins. They're studying monoclonal antibodies, biologics. And it's a way of determining if a protein is likely to aggregate at high concentration in a given formulation by examining its behavior in DLS at lower concentrations. It's a super cool application, and we actually have some webinars as well as some app notes about it.
That is definitely true. What is your accuracy and precision with antibody drug conjugates and labeled proteins?
So for our standards that we were using, so for standard purified samples, like what I showed you, we have accuracy within 2% and precision within 1%. But since ADCs are significant and labeled proteins get to be significantly more complicated, that 2%, 1% refers to the absorbance accuracy and precision. But a given sample, because it's more complicated, because there's potential for aggregation, might be slightly different.
Okay, can people select different absorbance ranges to quantify by ADCs?
Absolutely. So that's one of the great things about using homebrew. You can select whatever concentrate or whatever absorbance ranges you want, and do all kinds of other really neat calculations, including some stuff with DLS as part of that whole suite of homebrew functionality.
Okay. What if my drug absorbs in the same wavelength range as protein?
Sure. So yeah, that definitely can happen. A lot of drugs do absorb in that UV range from 220 or so to 30 up to 200 up to about 300, which is where proteins absorb. We actually have some customers who've done that, who have a drug that absorbs in that range and have still managed to use homebrew to quantify, determine DARs and look at antibody concentration and drug concentration. So it's just a matter of tweaking which absorbance maximum you're getting, and then making sure that you have extinction coefficients for all of your at your various wavelengths and for all of your various components.
Okay. How is this used differently for well, this as the stunner being for upstream and downstream samples?
Sure. So I mean, you can think of it. So for upstream stuff, for if you're just purifying out some bulk antibody, it's actually, you're going to be checking concentration anyway. And wouldn't it be nice to check and see if, there's some aggregates there as well before you move it on? I mean, that seems like it would be a really helpful quality check before you move it from your, that bulk purification farther downstream. And then for downstream people, if you're doing formulation studies, Stunner's super useful. If you're doing any sort of other process development stuff, which I guess could be upstream or downstream, depending on circumstances, also super useful. Really, anywhere you need to know about a protein concentration, or it would be useful to know if a sample's aggregated, that's where Stunner fits.
Okay. Someone is asking about the composition of the commercial buffer. I don't think we know that by heart. And then
yeah,
what is DAR and ADC? So
ADC stands for antibody drug conjugate. That's an antibody with a drug stuck to it. And then DAR is the drug antibody ratio. So that's the number of drugs you have per antibody molecule.
Okay, great. How do you put the 2 microliter sample on the plate? Is it possible with a Hamilton pipetter or a multi-channel manual pipette, or?
Yes to all of those things. It's super easy to load. You can use any sort of multi-channel pipetter. You can use any sort of, and people have used lots of different types of automation in order to load those plates. It's the same format as a 96-volt plate. If you can pipette into a 96-volt plate, you can pipette into one of these.
Okay. How big of an aggregates can Stunner measure?
So I believe that the upper limit is either 1,000 nanometers or 3,000 nanometers. Nels, can you give me a check on that one?
I'm fairly sure it's 1,000 indeed, but
1,000 nanometers. Okay, yeah. So no, 1,000 nanometers is our cutoff of size.
Okay. Couple of more. How large should aggregates be to show up as a separate peak in DLS?
So so aggregates need to be about 3 times larger in hydrodynamic diameter than your monoclonal peak in order to show up as a separate peak. So so 3-fold larger.
Yes.
And that's actually not just true for aggregates. That's pretty much anything. Anything, if there's a 3-fold difference in size, they'll show up as separate peaks.
Yes, there was a second part of it, how multimeric should it be, but with your three times the size, you pretty much answered it. And then I think we got to our final question. It's a bit a longer one. Can Stunner check aggregation based on the size of the particle by percent volume of various peaks?
So we use percent mass rather than percent volume, but they are very similar. So if you're used to using percent volume, percent mass gives you same information. And as you show you those on that mass distribution peak, and I think I'd have to check the reports. Yeah, but you can see it in that mass distribution peaks.
Yes, and you also get a percent of intensity for each peak in your report. So I think that would answer that.
That's what I was. Yeah, that's what I was trying to remember doing.
That's all the questions I have for you. So thank you. And thank you everyone for joining us today. I if you would like to have a deeper conversation with our team, please do get in touch with us at info@unchainedlabs.com or visit our website at www.unchainedlabs.com. We would, of course, love to connect with you. Thank you again for attending our virtual seminar, and I hope you have all a great rest of your day.